Everything seems dead and sterile, right? Wrong. These rooms are full of stories about life, but they hide in the freezers in the room next door. There, thousands of small pieces of animal bodies, the tissue samples, are stored at -80 degrees Celsius, each one of them labeled so they can be assigned to the original animal again. These lumps contain a myriad of stories about the diversity of life. But first things first, how did the animals end up in the freezer? Researchers catch the animals in the wild, with the needed permits, of course, and preserve them right away for scientific collections. They fix the body in formalin and place a small sample in a tube with alcohol or another preserving liquid. These are our tissue samples for the freezer in the museum. And now, the research in the lab can begin. First, the DNA, the animal's genetic material, has to be isolated from a sample. This is done with the help of a DNA extraction robot. On the one hand, by analyzing DNA meticulously, new animal species can be discovered and how they are related to each other. On the other hand, many animals are difficult to identify. But DNA can help determine the exact species an individual belongs to. One property of DNA is particularly useful. Every species has its very own distinctive DNA pattern. These patterns are created by the sequence of 4 different chemical bases, the building blocks that make up the DNA strands. An immense amount of information is stored in these patterns. The information can be collected using different examination methods. One of these methods is DNA barcoding. In barcoding, a small specific section of DNA is analyzed. In most animal species, these particular DNA sections have their very own pattern, just like a barcode at the grocery store. With barcoding, many animal species can be identified quickly and reliably. Let's take spider barcoding as an example. Before barcoding, some spider species could only be determined by examining a male spider. Only adult male spiders have large, noticeable reproductive organs in its head region, which look different depending on the species. Barcoding made it possible to reliably identify the species of female and especially baby spiders. Of course, this also works with other young animals, such as tadpoles or baby snails. But let's stay with spiders for a bit. Museum researchers have analyzed barcodes of many European spider species and have entered them in a worldwide digital barcode library. The digital library contains barcodes of countless different species determined by experts. This collection of barcodes is incredibly practical for fast and easy identification of animal species. Just analyze the barcode of an unknown animal and compare it to the barcodes in the reference library. Match. It is a yellow-vented bulbul. Speaking of birds, here's a story about owls. In 2013, bird watchers announced the discovery of a new owl species in Oman on the Arabian Peninsula. But museum researchers had their doubts. They analyzed the barcode of a previously known owl in the collection. And there it was. The new discovery was no discovery at all. It was a species that had already been described in 1878. But the barcode method also has its limits. The answer to some questions lies in multiple DNA sections, not just in a small fragment as the barcode. With further analysis of DNA, there have been many surprising discoveries. For example, a good-looking stranger walked into some researcher's trap in the Borneo rainforest. The result of the DNA analysis proved that the world had never seen this animal before, the red-hot chili frog. Or DNA analysis has shown, for instance, that very few switch points in a dog's DNA explain their diverse body shapes. Additionally, information about the environment is also stored in scientific collections. Ibexes, for example, have a record of climate change in their horns. Each year, layer by layer, the grass the animals eat leaves chemical traces in their horns. Analysis of these traces in old horns in the museum collection show that the plants save water and store more CO2 the warmer and drier it gets. The reaction of alpine flora to climate change is stored in these horns. Environmental pollution also leaves tracks in animals. In 2015, EPFL researchers examined 40 fish in the museum collection and found tiny pieces of plastic in their stomachs and intestines. This so-called microplastic comes from the millions of tons of plastic waste of every kind that end up in the water year after year. Water, sand, wind, and waves pulverize the waste. But it doesn't just disappear. It finds its way back to us through the food chain. Fish eat plastic, men eat fish. The research keeps on going, and we keep in suspense.